Excel has like weird calculations for everything.
Or for this class, we have like simulation things
Mhm.
built in. Usually.
Okay, that should be good now. So we're just picking up where we left off on Monday. So we talked about polymerase chain reaction very briefly. That's what the article covered. And there's a reason why we need to talk about this, because first, some of the other sequencing techniques use PCR or are based off of PCR, and you'll understand a little bit better. And that's also something that if you end up working in a lab that deals with molecular biology, you're going to do hundreds of those, thousands of those. So basically, it's an interplay of temperatures, timing, and with the mixture allows you to amplify a sequence of DNA. And you need to know a little bit of your sequence to be able to design primers, which are represented here, these two red things. I have two designs for this, so whichever one works better for you. So you do need to know a little bit about your sequence to design primers that will just be the start of your newly generated DNA. In your mix, you need an enzyme that will do the work and expand the newly generated sequence. DNTPs, which are your A, T, C, and Gs, and just a buffer that stabilizes the enzyme so it can do its job efficiently. And the first step is denaturation of DNA. But it's also, so that's mainly what it does, but it's also a step that activates the enzyme. And you'll see more on that slide. It says denaturation 95, 96. It's actually broader than just that. It's more like 94 to 98, depending on the enzyme that you are using. So if you do end up in the lab working with one of those enzymes that is called a polymerase, you will have to make sure that it corresponds to it. I'm just going to go back and forth so you can see both of those, whichever one speaks to you better. And so you see the temperature is here on the y-axis and time on the x-axis. So that is the first step, the increased denaturation of DNA and activation of the enzyme. Then the next step is annealing the primers. So it really depends on the primer sequence. Like the way you design it, usually you have some sort of control over this annealing temperature, but it depends on the primers, not the enzyme, not anything else, just like that. And in this case, the primers correspond to fragments of your DNA that you at least know partially, if not fully, implementation. But it will anneal to the template DNA that you want to amplify by sequence complementarity. So you remember we talked about A, T, C, G, and so each of those correspond to their complementary, right? And this is how they pair. And so your primer, let's say that's the sequence of your primer, will recognize the sequence of the DNA that is complementary. And so they bind weakly, but that's the signal that the enzyme needed to say, oh, there is a double-stranded RNA. I'm going to start doing my job and adding the building blocks to make a new sequence of DNA. And so there are then repetitions of cycle. I didn't say that. The next part is the extension, so that is adding the nucleotide to the sequence newly generated. Then this is going to be repeated 35 to 40 times typically. And so in the end, you're going to end up with a lot of your small sequence that is comprised between your two primers. And so you have usually a general idea of the size of your product. So it depends really on what you're trying to do. And that is the basis for PCR. Like there might be more details that you can ask me about if you have specific questions. But if you're interested and haven't already taken my class, we do a lot of PCR. So if you want to know how to do that, we do PCR. Then I just wanted to say too that in the end you end up with more of your product that is of that shorter size than your template. Then you had template in the beginning. So when you try to separate them on the gel, let's say you're going to find a big band of your product compared to your DNA that's going to get stuck in the oil by migration. So also other things mentioned that there are sequencing techniques that are based on PCR. PCR can also be used for the optimization of primer for downstream analysis or amplification for downstream analysis as well. So for Sanger sequencing, for instance, or when you're used to doing that for your next generation sequencing application, but it's to sequence in big numbers, a lot of reactions at the same time. We could use Sanger sequencing, but it will be a lot faster and more productive to use the next generation sequencing. And I'll get back to that in a couple slides. Then the technique for quantitative PCR is also the same. And we'll get back to that too in another slide. We just add a fluorescent dye to the mixture, and you'll understand why we do that. So PCR is really important. And one of the applications, nope, I don't know, sorry. It just says Sanger sequencing here. So it is based off of PCR. The only difference, or one of the differences, is that we're not only going to have dNTPs, so like your ATCGs, but also fluorescently activated, or trying to find what's fluorescently labeled nucleotides. So in addition to your regular nucleotides. So the primary annealing chain extension is going to go the same way, but depending on what nucleotide binds. So if it's a fluorescently labeled one, it will stop the reaction for this strand of DNA. And so in the end, after your 30, 40 cycles, you're going to end up with a bunch of different sizes fragments, because as soon as a fluorescent dye was added by the polymerase, it stopped the reaction. So that's what you have here. So here was at the first position, second position, third position. So you have to assume that these three were regular nucleotides, and then this one was added, this stopped the reaction, and so on. And so we're using a method of separation of those fragments by size, going through a capillary gel electrophoresis. So the fragments are going to migrate the smallest ones first, because they can migrate faster and easier through the matrix. And as they go through the matrix, a laser activates the fluorescence, and it sends the signal to a computer that records it in a ferrogram, and we know what nucleotide is at what position in that one. So they will migrate this way, actually. So that is the smallest, it migrates faster. The bigger ones are going to be at the top, and you're going to be able to read the sequence, but it's presented into this ferrogram on the computer. And so we constructed the sequence from here. Any questions so far? Okay. So Sanger sequencing, like I said last time, it's more for, like, if you're looking for a mutation in a region that you already know. And the key here is that you already have some information about your sequence. But when you don't, well, you get the next generation sequencing way. that is an oversimplification because sometimes even if you don't have information on your sequence, you can still work back from like a closely related species. Try to play with the temperatures, make the primers a little bit less stringent on what they're going to bind to, and you can still end up amplifying a product, send it to Sanger sequencing, and then you can redesign primers that are a little bit more stringent and more specific to your sequence. But let's say when we don't have a lot of information, you need to get it, or when we don't have any, you need to get it, and next generation sequencing is a way to do that. One good thing about it is that it goes pretty fast because there are millions of reactions going on at the same time in a next generation sequencing technique. And it's done on a chip or a flow cell, so it's pretty small. You only need a pretty small volume of your samples to work with, and yeah, instead it's fast. It's just a lot of reactions that I'm in parallel, so it goes pretty fast. Then I put a low cost, and I insist on it's relative because none of these are very cheap or anything, but it's becoming a little bit more affordable, let's say. But it's still depending on what coverage you want, what coverage you need to have a good confidence in your results. You can sort of adjust the costs there. But overall, if you were to do Sanger sequencing to sequence a genome, it would still be cheaper to do genome sequencing with next generation sequencing. That doesn't seem very cost efficient to use Sanger sequencing to sequence millions of nucleotides. So I just have 3 examples here. So genome sequencing, I just said you find information to assemble a genome, and that could be used for different applications I'm going to get back to. And then exome sequencing, transcriptome is more when you're interested in coding regions. And then targeted gene panel, it could be you're looking for pathogens in samples, so you're only going to amplify those pathogens and then you send them to sequencing. But it goes through using probes to really grab those pathogen sequences. These are the examples. Okay, one thing that you can do with genomic data, so obviously you can sequence DNA that you did not already know. You can assemble genomes. You can detect mutations, and at such a level that you can do population genetics, because mutations, like we said, some of them may be conserved because they don't really have an effect on the fitness of the individuals that are carrying them, and then they will remain in population. So it's great to follow migrating population, or even when you do those genetic testing, that's what they use, polymorphisms, to try to figure out who are your ancestors, or like what are your roots, but also who are your relatives. We use DNA sequences in evolutionary biology to try to figure out the sequence of evolution, the relationship between groups. And here I have phylogenetic tree of ABC transporters in insects. And so you can see there are different families, A, B, C, up to H. Like these ones are other transporters, but we can do phylogenetic trees using sequences for pretty much any level. Could be proteins like this, or it could be species, families, whatever. There are even people that do phylogenetic trees with languages and how they evolve, but that's not. We're using a sequence. So looking at a genome, like we said last time, we have access to information that we don't when we look at a transcriptome. So like regulatory regions, non-coding regions, So you get access to that. You can look at one gene, one chromosome, a full genome. You have different levels of focus depending on what you need. And you can do comparative genomics. So I have an example. It's the reference for the tree, but I have the reference for study there too using comparative genomics. Was a team I was involved in. We tried to that found. Explain that in simple terms. A way to control varroa mites using a sequence that does not exist in honeybees to be able to select their parasite in the honeybee so they remain unharmed. And I think I have a slide on that. Okay, I also wanted to bring this one up because it's not as well known of the general public. There are big units that do the sequencing reactions, but there are also portable units. And so this one is called the Minion. It's made by Oxford Nanopore Technology. And I thought that was cool because they brought that to space. You can bring that in the field. And if you have enough material, you can actually do the sequencing right there. And in some cases, it can be important. So like if you're responding to an emergency outbreak of a virus somewhere, it might be a good way to do that. And I thought that was pretty cool too, the technology. It also uses a flow cell, but it's not going to be just a chemical reaction that allows to determine what nucleotide is in your sequence. But here, the prepared sequence that you can visualize here is going to go through a pore in the flow cell, and the changes in voltage going through that pore will tell you what nucleotide it is. So different voltages. So not like Sanger sequencing where we use fluorescence. This is a little bit different, so I thought I would show you that. It's pretty cool. It's not as high throughput or as accurate as other technologies though. So depending on the level of accuracy and of coverage that you need, you might think that's not the right tool for you. Okay, so now I would like to talk about gene expression and how to understand changes during a stress response or uncovering differences in expression in a resistant strain to an insecticide compared to a susceptible strain, or how a situation evolves over time, how the expression changes over time. You would use transcript studies. So the ancestor of these are microarrays, and it's kind of like PCR. You kind of needed to know part of your sequence to be able to use that kind of like the primers, and then the rest of the sequence is added. So it only works on model organisms where you already have somewhat of a knowledge of some sequences of interest. And typically that's more used for human studies anyway. I don't know, it's kind of falling out of use too. Now we're using more RNA sequencing where transcriptomes are generated, and you can, like I said, look at expression changes due to stress, study pathways, study evolution over time of expression. The other way to study gene expression is by quantitative PCR. So here, just like PCR, you do need some prior knowledge on your sequence to be able to design your primers. Okay, so I'm going to walk you through what you have to do to first generate samples and then run them on a sequencer and then get your data for gene expression analysis using next generation sequencing. So you have your samples, so depending on the conditions, you're exposing two groups of insects to one insecticide, you want to know what it does to them, or different temperatures, different diets, you name it, then you can really look at a lot of different responses. Then you extract RNA, so there are kits for that, and then this RNA will be reverse transcribed into what we call cDNA. That's a complementary DNA that does not exist in eukaryote cells, but it really corresponds to the sequence of the messenger RNA that will just be transformed into a DNA form. Because I didn't mention that, but PCR, qPCR, all these methods, they don't read messenger RNA directly, they will read DNA. So DNA is always the template of PCR sequencing. So you have to go through that reverse transcription. And then that's also the time where we had tags, and that's what we call cDNA libraries. So then it can be processed from here, amplified during the run, and you end up with millions of short reads. So 100, 150 base pairs, 250 base pairs, I think 300 base pairs also happen depending on what library kit you're using, what instrument you're running it on. Then those millions of reads, you're going to have to align them to a reference sequence. So that's the complicated part. Like the beginning, okay, let's go. That's a lot of lab work, but then the instrument does a lot of the work for you. That's where you have to be somewhat proficient in bioinformatics. So I invite you to take my colleague, Dr. Pingold's class next fall, or maybe next spring or the spring after that, where she teaches you how to do some bioinformatic analysis, which is very useful if you want to go into that field. So, we just need to extract the information from the reads. That's what we need to do. And the first part is the alignment to the reference sequence. So how this works, we have to make sense of all those small fragments that, like, they are overly simplified. Imagine that there are 150 base pairs instead of just like 6. But it's just a representation. And this is your reference sequence. Could be a transcriptome, it could be a genome, whatever is available sometimes. And there's an algorithm, again, some of the packages that you use for the alignments that will be able to recognize patterns that are common, between the sequences, and with the overlap, you can reconstruct your sequence. I don't think you can actually do that here because that's really minimal, but you get the idea, right? This is what it really looks like. And I took the worst region possible because I have a lot of things going wrong here, but these would be the reads. And this is my reference sequence over here. Let's see here. And that's just one region. Imagine millions of nucleotides if you're aligning to a transcriptome, genome. You can really deal with nucleotides really more realistically. Okay, so how do you get the information that you need to understand gene expression from there? So your reads are aligned. You're going to be able to measure, or statistically, you can measure if there is a difference in the number of reads over the length of the transcript in your reference sequence between your two conditions. It's normalized by size. That's why I'm saying that. It's not just proportional straight or proportional. There's a function of size too. But to make it simple, the number of reads aligned to this transcript A in condition 1 and condition 2, is 2, 7, and here is 8. Statistically, it's not going to be different. Then if you look at transcript B between your two conditions, there's 6 reads aligned to the transcript B in condition 1, but there are, let's say, twice as many in condition 2. That is interesting, and that is statistically significant in this example, at least. But so that's how you sort of get information out of this. Because each of these reads, they come from sorry, I'm blanking. They come from what was in your sample. So if there were 10 transcripts of this transcript B, 10 versions of that transcript B, compared to the other sample where there were only 2, then it's going to be reflected in the number of reads. That's what I was trying to do for me. Okay, then you have this sequence, but you don't necessarily know what it is. So you have to go through an analysis that will tell you what is the most likely identity of your nucleotide sequence. Sometimes you can translate it into a protein sequence, so like it's useful when you're working with protein regions. So I put an example here. So I use a lot BLAST, and there's a tool called BLAST to go for bigger data sets, so typically RNA sequencing data, after you've done the analysis. And you can input your nucleotide sequence. So I put this one here as an example into BLAST, and it generates alignments that's telling you what is the most likely identity of your sequence. And here I use the BLAST X tool. So this one will translate your sequence and look for the protein identity and how it aligns, how it compares to the other proteins in the database. Okay, so I picked a cytochrome P450 on purpose, but sometimes you don't know, I don't know how that is. And I don't remember how many alignments were generated, but then you're going to look at them separately. So you're going to look at the top ones, first of all, because they are the closest to your sequence in identity, in just alignment, complementarity, whatever you want. So there are different scores that you can look at. The e-value, if it's closer to 0 or equal to 0, then it's a good thing. Percentage identity, so that's how many nucleotides are the same in your sequence and the sequence in the database. So the first one is 100%. So there's a pretty good chance that it is your sequence. But there's only 65% cover, so that means the sequence in the database is longer. And what do we have? Yeah, the accession number is in this case, XP, it's a protein in the database, so it sends you to the protein that it was aligned to. In this case, how the translation of your sequence was aligned to this protein. And so you can look at the differences in the top 10, or depending on what the data here tells you, you can go lower, you can just look at the first 2 ones or first one. But you can see that there are differences in nomenclature. So this first one that is 100% our sequence, because I took it, like that's the sequence, is called P450-9F2, and it comes from Aedes aegypti. But then if you look at the second match, it's called cytochrome P450-9J2. So there's a little bit of a question there, but depending on how deep you want to go. Like you might be happy just knowing it's a cytochrome P450, or you might need to do a little bit more research on which one is most likely, which one has the most chances to be involved in your study, that kind of stuff. Or you can go back to the sequence, really look at it, determine if there are really any difference between the 9F2 and 9J2, or it's just a question of nomenclature. I admit that I was looking for a 9J and then this one popped up. I was like, that's strange. Okay, so any questions so far? Okay, you get a list of your transcripts that are differentially expressed after you did the statistical analysis of your aligned reads. And then you can proceed to like pathway analysis, conserved domains, you name it. There are a lot of things that you can do, that network analysis. Yeah. But that is for a more advanced class, not this one. Okay, I would like to talk about qPCR too. So it's quantitative PCR. I have reverse transcriptase in parentheses because I want to make sure there's no confusion. We also call it QRT-PCR or RT-qPCR. Some people started using RT as real time because you can follow the amplification in real time, but that's not what it stands for. It's for reverse transcriptase because you're starting from messenger RNA. So you got to bring it to the DNA form that will be recognized and used as a template by your polymerase enzyme. So just wanted to bring that to your attention. It's the same principle than PCR. So you have those cycles, denaturation, activation, annealing of the primers, adding the building blocks, repeat that 30, 40 times, and then yeah, that's the end. The only difference is that you add a fluorescent dye. So here I cited cybergreen. Depending on the study, you may look at different dyes, but that's the most widely used for gene expression. And you can follow in real time the amplification of your product of interest because at each cycle, it records the fluorescence level and it generates this curve here, which is the amplification. So at the beginning, you have very little fluorescence. But as your product amplifies, as the fluorescence accumulates, it will go through this sigmoid amplification here and then reach a plateau because it can't amplify more. So how you analyze this data really is based on what we call the cycle threshold value or CT value. The CT threshold is determined the same for all the samples that you're analyzing. Here they're represented in different colors. It's easy, but when you look at actual graphs of the amplification, they're all the same color, so it's really, you know what your sample is. You do need to know that. And the cycle threshold is, it's going to be the value that you use for the comparison. So let's say this first curve here is your susceptible strain of insect that is your control. And then your orange curve, that last one, is your resistant insect. And you should have replication for each, but let's make it simple. So if you compare through the delta-delta CT method or other methods that kind of work like ANOVAs, you can determine if this CT value is different statistically from this CT value. And just looking at that, there's a pretty good chance because we're talking about numbers of cycles here. So the more, the amount let me try to find that because it's not necessarily super straightforward, but the more sequence you have, the faster it will amplify. So your CT value is going to be small. So typically, genes that are expressed in all the cells, like actin or trying to think of the ones that I use, GAPDH, stuff like that are structural, expressed in all cells at all times, you'll have a very low CT value because there are just a lot of them. But then a gene that's not highly expressed or only expressed in some conditions, you're going to have a higher CT value. And so that's kind of what we're seeing here. This one is very abundant, so it crosses the fluorescence threshold faster than the one here that's less abundant and will cross the cycle threshold, the fluorescence threshold, much later in time and in numbers of cycles. So that's what we're comparing. What else can I say to help you? You do need statistics to figure this out, calculations. It's not as straightforward as it looks, but like when you have two curves that are very far apart from each other, there's a good chance that your samples are going to be different for that gene. Okay. Any questions? Same thing in my class. So that's just an example from one of my publications to kind of tie back your cytochrome P450-9J with gene expression, what we talked about. So here I looked at the expression in the susceptible and pyrethroid resistant strain of Aedes aegypti, yellow fever mosquitoes, and I looked at males and females at different ages. So these post-emergent as adults, and they were not treated. That is just their constitutive differences. And so these three transcripts, 9J10, 19, and 28, were statistically flagged as significantly different. And you can see here, like if you look at all ages, both sexes, it's much higher in the resistant strain here too. It's also much more consistent between the two sexes. And then this one is very, very obvious because it's almost not expressed in the susceptible strain and it's highly expressed in the resistant strain. So you have different levels of significance, different levels of expression in the same samples. Just to show you an example. What that looks like. Why do I have this? Oh yeah. Okay. So you can also use qPCR for other things than looking at gene expression. You can detect and quantify pathogens, for instance, or bacteria from the microbiome, and we're just using basically the same idea. The CT value is going to be what you're interested in. So sometimes you just want to know if your sample has the pathogen that you're looking for. Again, we're used to doing that. Where the amplification is going to be pretty obvious in the middle of a bunch of negative samples. You're going to have one that has that sigmoid curve. Oh, this one's positive. Depending on where the CT value is, where your curve is going to cross that threshold, it also gives you an information of how much of your pathogen there is in the sample. So I thought I would mention that too. Okay. Just quick, I want to check on the. I'm going to be pretty brief here. So we talked about regulation, like the gene expression depends on condition, environmental conditions, on response to stress, on response, like just the differences between strains, all that. But there is a mechanism that's becoming trendy, more obvious, very important to understand, it's epigenetics. So that's a mechanism that allows from one genotype to have many phenotypes in response to the environment. Let's keep it very broad. But it's heritable. So that means somehow it's in the DNA. So if we look back at the DNA, it's the structure of the DNA that dictates gene expression regulation in this case, not the sequence of the DNA, if that makes sense. So that occurs with an interplay of methylation, demethylation, acetylation, deacetylation that modify histone proteins. And just so you know, methylation, acetylation, you might have already seen that if it's attached to a protein. What is that?
Acetylcholine.
Yeah. But in this case, we're not. That was just a side note. But yeah, that's what that looks like. These groups are attached to the DNA methyl acetal groups, attached or detached. And so the interplay between these two, the differences in activation of the promoters will change if a gene is expressed or not. I don't know if I'm explaining it really well because I kind of added that yesterday and I didn't really have time to digest the slide. But I invite you to look for yourself what that means. There is a chapter in the book. If you don't have it, I can just send a copy of it. But yeah, it's in the book. There's a whole chapter about gene expression and epigenetics. So basically, the histone protein modification helps insects adapt to environmental changes such as diet, temperature, even caste selection in honeybees. Yeah, that's at the DNA level. So that's pretty important if you think about it. So these are just, I tried to have representation of what it is. So these are your histones when they are demethylated. Then if you add a methyl group to it, and that's through the methylase protein that can do that, which needs to be expressed as well, just as a side note. You can activate or deactivate the expression of a gene that is downstream. As simple as I can do, I think. Okay, I wanted to go a little bit deeper on how we can actually use that more on the pest control or insect protection lens. And so I wanted to talk about transgenic plants, for instance. So that's just an example of BT because it's been widely used in crop protection, and that was something that I worked on. So dear to my heart. I suffered a lot with that. So you can have a plant express a protein that is not itself, it's not its own. And that's basically what transgenic is about. So I mentioned cryotoxins, they come from a bacterium, it's called Bacillus thuringiensis. And so it's just having the gene that encodes Bacillus thuringiensis because it's a protein. The gene that codes cryotoxins from Bacillus thuringiensis inserted into the genome of corn in this case. And so the technique, how they do that, it doesn't matter because they change over time when they find new, easier ways to do it, more reliable ways to do it. But basically, the gene needs to be added with a promoter or using a clone promoter, it doesn't matter. But basically, end of story, the gene will be expressed like it's part of the corn genome. Oh, that was painful. Let's not talk about it anymore. But yeah, so transgenic, that's how it works, basically. You take a foreign sequence to have it expressed in the plant. Whatever. Then there's another example of transgenic plants that express RNAi traits. For example, the SNF7 trait in corn against Western cone rootworms. But I also am taking advantage of this slide to talk about RNAi used in non-transgene forms, but I think it's a spray. There's recently acaricide targeting Varroamite specifically that was commercialized to eliminate them from honeybee colonies. So I'm guessing you know how RNAi works. Kinda. Okay, you should have seen that in the immunity chapter because it's a innate mechanism that invertebrates have to recognize a viral attack, a viral invasion, and break it down right away. So how they do that is because a lot of viruses have double-stranded RNA as a genome, and eukaryotes don't have double-stranded RNA. So the machinery will recognize that is non-self and attack it. So the machinery consists of a complex that's called the RISC loading complex, made of 2 proteins, and they will recognize the double-stranded RNA as being invading the system, invading the cell, and dicer will cut the double-stranded RNA. And one of the 2 strands of that double strand will be used as a guide to go find other representatives of this small sequence that come from the virus to eliminate it by cutting it. And so there's another protein that's involved called organote. But so what it does is, go find the other virus molecules that could be invading and cleave them into small sequences. So in the end, the virus is not able to use the host machinery to replicate itself. Scientists are very resourceful people, and we found a way to use it for first study the function of a transcript, function of a gene, but also to study to kill pests. So how it works, instead of a viral double-stranded RNA, here we generate an artificial double-stranded RNA that actually corresponds to a transcript of either the transcript of the gene we want to understand what it does, what the function is, or a vital gene that will kill the arthropod in question. So it could be, like I mentioned, the SNF7, that's a gene that is expressed in lysosomes, subacular ATPase. That's a pretty important mechanism in the cell to get rid of what's not wanted. Also transport within the cell. I mean, it is vital. So in that case, it's if you don't have it as an insect, you die. The Varroamite trait, I don't remember what it targets. No, I think one of these 2 paper might be it. So what else can I say? Yeah, the double-stranded RNA that we give them, the feeding, injecting, there are different ways that they can receive that synthetic double-stranded RNA. The complex will recognize it as non-self, use one of the strands as a guide, go find the complementary of the guide strand, destroy the messenger RNA in the cells. And what we have in return is a decrease in expression of that. And so the protein cannot be expressed. Proteins are structural. We can figure out if something is wrong, if something is it changes color, or if the insect dies. It could even be, let's say, if we think back to resistance, we could try to knock down one of those cytochrome P450s that are overly expressed in resistant strain to try to restore the susceptibility to an insecticide. An application is just not one, so it might be a little bit more complicated than that. But in theory, that could be. I still don't remember what's that. No, I forgot. I'll get back to you. That's just another representation of it. If that speaks more to you. It's a little bit more detailed. So yeah, the consequence of this, the messenger RNA is degraded in the cell, so it's not going to encode. Okay, there's another mechanism. You might have heard of it more in the medical field, CRISPR-Cas9. It's actually CRISPR and different type of enzymes here, but that stands for clustered regularly interspaced short palindromic repeats. And basically it's targeted genome editing. So that mechanism is in bacteria. And it's also an antivirus and phage system that we could call adaptive immunity. So it works a little bit like RNAi, but the difference is instead of a double-stranded RNA that is matched to another RNA that is single-stranded or double-stranded, if we're thinking about the natural occurring mechanism, here the RNA is matched to a genomic DNA sequence. And so doing that, yeah, it's not double-stranded RNA. It's matched to no, whatever. It also uses a guide RNA, but the target is going to be a genomic DNA sequence. And so the same way, it's going to recognize the guide and try to find where it occurs in the genome, then cut that part out, and it can be replaced by another one. So you can either correct or replace sequences that may be causing diseases, causing resistance, or you can disrupt that sequence by adding or removing parts of it. And like if you think back to Monday, frame shift, mutations, insertion, deletions, then you change what the sequence does, what protein ends up being translated from it. And we can also use that to figure out what sequence does in the genome. And that's another representation of it. Okay, I think we have very few slides left. So that's the last one I wanted to talk about. I did mention comparative genomics earlier when I was talking about what we can do with whole genomes. And I just wanted to mention that study that we were both involved in, but we didn't do the fancy parts. So that is trying to find a target for varroa mite control without affecting honeybee health. And they found out that there was protein receptor that doesn't exist in honeybees, but that is present in varroa mites. So you can see varroa mite here, varroa distractor, and that is where the receptor is found. These are all the groups where the receptor exists. And the other ones don't have it, including Apis mellifera, the honeybee. So by synthesizing different short peptides that can go bind to that receptor, you can disrupt the physiology of varroa mites to a point that they have muscular responses that are detrimental to them, basically. Yeah, so if you're interested, you can read this to get more information about the sequencing part, because the rest is more testing if they work or not. Okay, so let's conclude. There are different things I would like you to take away from this double lecture. So genetics and physiology are directly linked through the central dogma. And the methods to understand this relationship, DNA, RNA, proteins, or to get information from each of these levels are becoming more affordable and more widely used. So there are resources out there for the analysis, particularly, and protocols to get the best libraries possible. Then the genetic data can also be used to our advantage in crop control, beneficial insect protection, understanding the function of some genes. And I don't know, I just put two examples. One of them I really like Culex pipiens ABC transporter study, because they use a wide array of techniques to figure out what ABC transporters do in the cuticle or in the cells below the cuticle. And they used chemistries to confirm that ABC transporters were involved at all. Then looking more specifically at the sequences, at the transcripts that were expressed in the legs of mosquitoes, they found that these a few ABC transporters were particularly expressed in that part of the body. They shut them down with RNAi to verify what happens when these ABC transporters are not working. And there were effects on penetration of an insecticide, but also the structure of the cuticle. It was much thinner and also somewhat misshapen. So ABC transporters are important for the formation of the cuticle in the legs and penetration of the insecticide in contact. So I thought that was pretty cool. And that's one of my interests and my research interests are ABC transporters. So I thought I would put it there for you. The other one is more how Plasmodium falciparum, the main agent of malaria, is becoming resistant to some treatments. I don't remember which one right now, but through epigenetic changes. So the study itself might be a little bit complicated, but I think you can get the gist of it if you read it. And then I wanted to end, shameless, I'm promoting my class in the fall. I think five undergrads already signed up, so if you're one of them, thank you. And yeah, we're gonna cover more the hands-on part of what we talked about here, trying to understand what is going on when an insect is exposed to an insecticide, or I might have some microbiome samples to where we can look at what bacteria are there or not. We're going to do a lot of PCR extractions of RNA, DNA, and gels, primer design, and if we're lucky, Sanger sequencing analysis. We're not gonna do the Sanger sequencing. I don't have the instrument, but we're gonna look at the sequence. So yeah, those of you who already took it, you don't care anymore, but I would end on that because that's the hands-on part of it. Yes.
Okay, so Cas9 is used for kind of adaptive immunity in bacteria, but could it also be used like that in eukaryotes, either by, I don't know, having it being like, here's some Cas9 for this virus, or like injecting that into the genome with the ability to make its own proteins.
Oh, I see what you mean. More like the transgenic part, you have them express a CRISPR-Cas9 or CRISPR and whatever Cas phase it is, and I don't know if it's one. I mean, interesting. I didn't think of that. I don't know how that would work though. I mean, there are ethical concerns in doing that. But also I'm not sure if the organism would be sustainable, because if it's targeting its own mechanisms, I guess it depends on what is the guide sequence.
I was
That's a good
thinking
question.
like a guide sequence for a virus.
Yeah, so maybe it would be more used like it is used in bacteria. Like yeah, to recognize virus invasions and break them down in phage, break them down. That's really, yeah, I don't know. I haven't really thought of that. Yeah, I think it depends on the guide sequence. In theory, sure, there are proteins, you can express proteins. You know, there are controversies about using CRISPR-Cas9 in the medical field, depending on what type of cells are targeted. So somatic cells is acceptable. Germ cells not, because there is this whole idea of you don't know what's wrong. Like it will affect the next generation, basically. So I know there's been ethical issues about that. So I think right now it's only used for somatic cells. But I'm guessing it has a very limited efficacy because yeah, if you have a genetic mutation that you don't want to pass on to your children, then that's why it would be interesting to modify germ cells, but in the sense of if insects expressed the CRISPR-Cas9 system, yeah, I don't know how much different it would be compared to RNAi. Yeah, I don't know if that would bring anything to them extra.
It could like maybe interrupt some of the more like propagated viruses, like for like thrips, for example, like the virus has to multiply in the thrip. That could maybe interrupt that and prevent them from transmitting.
Maybe. I mean, I was thinking about it now because I believe RNAi and C. elegans can be systemic and pass on to the next generation, or at least the effects from it.
Yeah. Yeah, so there is a whole thing about parental RNA. You treat the parent and the effects are visible in the offspring. Yes. Bobby would don't have the same ethical issues here. Just saying. Yeah, once again, I guess it depends, on what is targeted by the mechanism. Single genomic RNA, yeah, it's non-self too, but is it able to tell that it's not self? If the mechanism is not native, that's what I'm wondering. What would be recognized as a threat and what would be?
Yeah, like when you give it, would it almost have an autoimmune disorder by giving it Cas9?
What traits do you have?
You know, I would encourage you just to parallel the Carrie Mullis paper that I provided. I would encourage you to read about the work of Jennifer Nunda from UC Berkeley, who discovered CRISPR-Cas9 technology, won the Nobel Prize for it long ago because of the work for COVID therapies using this technology. There's a bit of competition on who actually discovered it, so. All right, any other questions? Okay, just keep in mind, concept notes are due in basically 20 days. So I will accept them earlier if available, but don't rush. Also, I will put the final exam on Canvas on Monday. Please take your time with it. You can have longer than a week. Okay, I know that there are a lot of assignments that are coming up. It'll have a similar format as the last one, so hopefully it doesn't take you more than an hour. And just keep an eye out for other announcements. All right? Good rest of your week.
Monday, finals week or Monday? Which Monday? Next week? Okay.
The upcoming